Good morning, everyone. My name is Gao Xin. I'm from Bioinformatics Group of Computer Science. The title of my project is Bioinformatics in Health Informatics. I will focus on protein folding problem and prion diseases. Today, I want to talk about 5 parts of my project. First, I will give a short description of prion diseases in health informatics. Then, I want to talk about protein folding problem in bioinformatics. And there is a short review of the work on prion diseases and current researches in protein folding problem. And I will also talk about some issues and possible solutions. The first prion disease discovered was scrapie. Scrapie was first found in Europe in 17th century on sheep. And in 1950s, Carleton discovered kuru in New Guinea. Kuru attacks the neural matter of brain, and it can cause infected person to die within 3 months. There are also 2 other kinds of famous prion diseases. One is BSE, which is famous for mad cow disease. And it's a kind of nerve center system pathological change on cause. The symptom of BSE is very similar to that of scrapie. And the other disease is VCJD. It's a rare and fatal disease on humans. Professor Stanley Persiner of University of California, San Francisco proposed the link between these 4 kinds of diseases. He said all of these diseases were caused by a kind of protein, which is called prion, and he was awarded the Nobel Prize in 1997. But until now, no one can prove that Persiner's theory is right, so it's full of suspicions here. This picture shows the incidence of BSE all over the world. We may see BSE and VCJD are severe in some parts in Europe and also in Canada. Now, what's the relationship between prion diseases and health informatics? Well, prion disease is a very important part in health area because it is closely related to many domains in health area. For example, biological science, medical science, research and education, prevention, promotion, and population health. Now, how can prion proteins cause diseases? Well, people now know that prion proteins have 2 forms. One is Prpc and the other one is Prpsc. Prpc is the normal form of prion proteins, which can exist in the body of everyone. And Prpsc is a bad form of prion proteins. It is pathogenic. This picture shows the structure of Prpc and Prpsc. This kind of structure is called alpha helix, and this kind of structure is called beta sheet. Both alpha helix and beta sheet are kinds of secondary structure of protein structure. So we can see the difference between Prpc and Prpsc is there are much more beta sheets in the structure of Prpsc. Now, from the study of prion diseases, people now know it is the possible reason for prion disease like this. Prpc can make Prpsc to transform to the bad form of prion proteins. And then there will be more and more Prpsc accumulating in nerve cell. Then it can kill nerve cell, so make the brain tissue spongiform. People now know Prpc and Prpsc have the same amino sequence, but the 3D structures of them are different. So we guess Prpsc is the result of prion protein misfolding. What is protein folding? Well, protein folding problem is given the sequence of amino acids of a protein. For example, here, we want to go through some particular process, then get the 3D structure of this protein. So protein folding problem is very important for us to know about the function of this protein. And it is also important to design experiments to improve stability of this protein. It's also very necessary for drug design. Now let's take a look at the work on prion diseases. From the study of prion diseases, people now know there are probably 3 causes for prion diseases. First one is transmission, which means one can acquire a defective prion from the outside. And this is also considered to be the main reason for VCJD on human. And the second reason is spontaneous, which means one's own prion proteins can turn to the disease form. But in September 2004, Bruce published his paper, A Fresh Look at BSE on Science, and he said that this was the least likely reason for prion diseases, because although we know prion proteins can turn to the disease form, but the time for this process is much longer than 50 years, which is much longer than the whole lifetime of an animal. So it is least likely. And the third cause for prion disease is genetics, which means if parents have prion diseases, the children will have a higher chance to have prion diseases. And people also have explored the sequences of prion proteins. What they do is that they do a multiple alignment on different kinds of prion proteins from different kinds of animals. Then they use some particular scoring function, scoring scheme or coloring scheme to get the important part or the conserved part of prion proteins. So in this way, they can know which part of prion proteins is conserved well and is important. And there are also some researchers in Japan that we can make some kind of proteins to prevent the brain nerve tissue. The way they do this is they try to bind this kind of proteins to the bad form of prion proteins. And they make this binding just have physical activities without chemical activities. So in this way, although they cannot eliminate the bad form of prion proteins, they can prevent them from infecting people. And this guy in Japan says, since Prpsc is a result of prion protein misfolding, so we can try to unfold it first, then control it and refold it in a different way and make it to become the good form of prion proteins. Of course, there are a lot of researches on protein folding problem. Typically, people have two methods to do protein folding. First one is experimental method. For example, X-ray crystallography, NMR spectrometry. But all of these experimental methods are highly time-consuming and money-consuming. And the other method is computational method, which is protein folding prediction by computer science. This picture shows the relationship between prion diseases and bioinformatics. We can see computational protein folding method is a very important part in both prion diseases and in bioinformatics. And of course, there are other areas which bioinformatics can be used in prion diseases, for example, drug design. Now, what can computational protein folding method contribute to prion diseases? Well, I think there are many possible uses here. For example, with this computational method, we can predict the structure of different prion proteins, so we can understand the function of prion proteins. And we can analyze the conformation of prion proteins and use it for drug design. And we can also do some experiments on computer. So in this way, we should save a lot of money and time. And if we know the process of prion proteins folding and misfolding, then we can use computer to simulate this process. And of course, it is possible we can find some treatment for prion diseases from the known treatment for other kinds of diseases. Now, what can we do for protein folding prediction? There are typically two methods. First one is ab initio prediction, which means we have to predict the structure of a protein directly from original sequence of amino acids. So it can deal well with the short sequence length. A famous program for this is Rosetta, which is produced by David Baker in University of Washington. And the second way is protein threading. For protein threading, we have a database of proteins with known structures. Then what we want to do is we want to match the target protein to each one in the database and then pick the best one. A famous program for this is Raptor, which is produced by Jinbo Xu and Ming Li in University of Waterloo. This is a screenshot of Raptor. We can see Raptor can show the structure of protein well, and of course, the prediction performance for Raptor is also very good. On the other hand, there are a whole bunch of issues here on both prion diseases and protein folding problem. For example, there are some issues about protein folding problem. the basic assumption for protein folding problem is a protein has one and only one stable status. But from the study of prion diseases, we already know prion proteins have two stable forms, which are Prpc and Prpsc. So how to deal with that? The possible solution may be, for example, this is our energy function. We have a lot of local minima here. Now we want to do is just want to get this global minimum. So to deal with this issue, we can try to get all of this minimum here rather than getting just this local minimum. And the target for protein folding problem is we have a particular energy function, and we want to minimize this energy function. But it is known that some proteins do not fold the lowest energy conformation. So what can we do? Well, although these proteins do not fold the lowest energy conformation, they will probably fold to the one which is close to the lowest energy. So we can allow some possible errors on the energy function. And there is also a dynamic issue here, which is a protein will undergo some small dynamic changes in conformation. Should this be modeled? Well, I think we won't have any good solutions for this until we are able to observe and understand the process of these dynamic changes. And there are also some issues about algorithms. For example, protein threading is a simple part of protein folding problem, but even protein threading is proved to be NP-hard. And we don't know how to get the most reasonable energy function. We don't even know how to evaluate the results of our algorithms. And of course, all of these algorithms are highly time-consuming. And on the other hand, there are some issues about prion diseases. For example, we need to know the function of the good form of prion proteins. And we also need to know how bad form causes diseases. And we need to understand the conversion process between the good form and the bad form of prion proteins. So we can see there are a large number of issues here. And of course, a lot of challenging problems need to be solved, and a lot of chances here. So that's my presentation. Thank you. Any questions or comments?
Question. Just towards the end, you mentioned that it's computationally difficult to process these problems. Is it to some extent just a matter of waiting until the computational power is there, until processors are faster and or cheaper, or more complicated than that?
Sorry, could you see?
Explain NP-hard.
Oh, explain NP-hard?
No, it's just a matter of waiting for the processors to grow in the power, or what does NP-hard mean?
Oh, NP-hard means we won't have, NP-hard is a class of similar problems. And if we can solve one of them in polynomial time, then we can solve all of this class in polynomial time. It means we probably cannot solve it in polynomial time forever, but it's not for sure. So is it clear?
Yeah, no, I understand that. I was just wondering if processors were 10 times faster, would that ease the problem, or is the magnitude of the problem so great that it doesn't matter how fast the processors are?
If you had a computer 10 times faster, would that help?
Oh, actually, that will help a lot, but that will not solve this problem radically.
I have one, and it could be my misunderstanding, but I thought one of Prusner's claims was that the introduction of the diseased form of prion into the material of the, let's say, the brain, actually caused other prions to misfold. Is that not proved? Is that one of the issues?
Yeah, and that's a lot of controversies here, because some researchers think the only prion theory of Prusner is totally wrong, and some researchers, but I think more researchers believe Prusner is right, and there are a lot of controversies here about the reason for the real prion diseases. So it's not for sure.
I was just reading in Science about an experiment in the last few months related to this, and where there was at least reasonable evidence that the introduction of a prion into an animal caused a rapid onset of, I can't remember which animal it was, but essentially prion disease anyway. So there's at least some early result that is beginning to come close to showing that the infection happens. The question is, if you introduce a small number of prions, is there evidence that that causes other prions to form from the normal prions in the brain? But you're saying that's not known yet.
Yeah, okay. I think that's not known yet. Hi.
Just out of curiosity, could you show the figure again of the world map in which
The world map?
Oh, okay. This one?
So I was just wondering if this has anything to do with UV radiations, ultraviolet radiations, because the regions that I can see is the ozone layer is quite depleted in these radiations. So I was it's out of curiosity if it has anything to do with these.
So the question is there any relationship between ultraviolet radiation from the sun and the location of these the highly prevalent BSE, for example?
I don't think there's any relationship between them.
I don't think you'd find one either, but I don't know. I guess your theory would be that somehow the energy of ultraviolet radiation was a trigger. Have you read something that there's...
No, I haven't read anything, but the regions that I see, like the Canadian region,
Ah.
I guess was on depleted areas of western of Canada and then towards western Europe as well. So that's why I'm just curious if it has anything to do with that. I can't see Australia.
Please?
I had a question. Like typically, like in molecular biology, the sequence of like DNA that gets transcribed to protein,
Yeah.
it's like one process, and that the sequence is the actual code, right? Because this is like a very big anomaly then, because if the sequences are the same between the good form of prions and
Yeah.
the disease form of prions, and this is a big hole in the theory of the genetic, like the fundamental theory of the genetic model, right? Like that DNA gets transcribed to RNA to protein. So wouldn't that suggest that there are other proteins probably interplay with prions? Because if they're misfolded, like the amino acid sequence should tell the cell structure how to fold the protein.
Yeah. Most people believe that there's some hyper things to help prion to do misfolding or folding. And there are probably some kinds of catalyzed reaction for this process.
Okay. Yeah, basically, the cell does not tell the protein how to fold.
Yeah.
Physics does. But the conditions of the cell can determine the types of folding that occurs. Is that a good statement?
Yeah.
So it's not programmed into the cell. It's more programmed into the protein.
But like, because if the sequences are the same, then like I mean I guess the whole what you talked about, the local and global minimum that in energy states, but like the different like chemical bonds that would form between the amino acids and between the proteins, amino acids would should theoretically fold into one configuration.
Not necessarily. Your hair doesn't do that. Your hair.
Frizzy days, yeah.
Yeah, that's right. I mean, according to the conditions, the folding can occur differently, or the shape of the protein.
Yeah, because there are a lot of factors can affect the final shape of this protein.
Particularly the ionic environment is one of them. That's a little factor. The other thing is this folding occurs, you probably correct me, in nanoseconds, I believe.
Yeah.
The folding is a very rapid process that's, that occurs. I was quite amazed by it. And so it's pure physics. Thank you for an excellent presentation.
Thank you.